carbons, reds being oxygens, blues nitrogens. So you should be able to see those things in there. All right, so what's going to happen here? So we have our beta subunits. You can see that there, you can see that tetramer here. You can see that there are the 4 subunits. We roll it around. There are spaces between. You can get the feel from it. I've rendered it so you see the surface, but if you look carefully, you can still see the secondary structure embedded behind that. Now to make that a little more obvious, we can always go up here to the settings and we can change that. Transparency, go to surface, and let's go ahead and make it about 50%. So now you can see that we don't see the surface quite as well, but it's still just a ghost of a structure there. All right, get a feel for what it is. Now what I'd like to do is point out a few things today. One is, if we're looking, if we want to go ahead and zoom in here, let's see, zoom in is this one. So if we're looking at our porphyrin ring structure, what do you notice above and below that porphyrin ring structure? We see a pair of histidines, correct? Can you identify which one is probably distal, which one is probably proximal?
Yes. Proximal on the left, distal on the
Proximal is the one on top to the left. All right, you can see the distance between these is a little closer, and you can see that the distance between the distal one is a little further. The other thing I want you to take note of is there's no ligand bound right now, correct? And so when there's no ligand bound, see if I can rotate it ever so slightly, you see that the ring structure itself of the porphyrin is not exactly planar. The iron is actually to the side of the proximal histidine. There's not, that's losing the tug of war, all right? This side, histidine's winning, this side's losing. If oxygen were to come in and bind, then we can actually see that that would shift into the plane, right? So that's something to take note of there as well. Let's see. And the reality is, if we shuffle on over here to a different one, I didn't label that one. Where's the other red one? Oops. Right, zoom out, and red over there. All right, so this one I just did 2 sets of them is what I did. So now if we zoom in, you can see a similar thing going on with this particular structure as well. So we have the distal and we have the proximal histidines. Any questions so far about structure itself? All right, so the thing we need to talk about today is called the Bohr effect. What's the Bohr effect? Who knows? pH in the body, what?
pH in the body, like tissues in the lungs differ. So
Okay. Okay, so the pH differs between the blood and the tissues, and there's a difference in affinity that was observed by a dead scientist named Bohr. All right? It's actually Niels Bohr's father. Remember Niels Bohr? He was the one responsible for the model for the atom of hydrogen. Anyway, so his father was actually a physiologist, and he's the one that described this phenomenon, that with the change in pH, hemoglobin will actually alter its affinity for oxygen. Now the question is why? So if we're thinking about it for a moment, what's different in the tissues versus the lungs? We'll just go there. What's high in concentration in the lungs?
oxygen.
Okay, so oxygen concentrations are higher, the partial pressure of oxygen is. And then in the tissue, the partial pressure of CO2 is increased, correct? All right, so with that, what's interesting is the hemoglobin will come through up here and it'll be a high affinity for oxygen. What do we call that high affinity state of hemoglobin? The R state, that's right. So the R state is predominant in the lungs, and then from there it'll transfer down into the tissues, and it will turn into what state? The T state. All right? And then of course that can go back to the lungs and reset. So that's the basic process we're looking at. Now, Dr. Bohr said that the pH was one of the factors that would affect the affinity. What else did he say affects it? What was that? I heard a faint voice. Temperature. Temperature will affect it, but not in the same fashion as these other ones will. But definitely temperature can. It turns out that it's the carbon dioxide concentration itself that influences the state. So, high concentration of CO2 here, lower concentration of CO2 up here where the oxygen's high. Now, why would CO2 and pH be related is the real question. Let's go with those. All right, so what happens to CO2 if it dissolves? We're gonna have H2O come in, dissolve it, it's gonna turn into carbonic acid, which then, as you learned in 106, being a weak acid, will do what?
Create a buffer.
It'll create a buffer, which actually is very useful to us. And so it can dissociate, forming bicarbonate and our hydronium. All right, so that's what we have. Sorry for the monitor being away. All right, so that's what we have going on here. So ultimately, a high concentration of CO2 will lead to an increase in a hydronium ion. If there's an increase in hydronium ion, what happens to the pH?
It decreases.
It decreases, yeah, it's that dumb negative log thing, right? Inverted. So, all right, so basically, his observation was one and the same due to the equilibrium effects of carbon dioxide being hydrated. Now, carbon dioxide does not hydrate very well on its own in water. For evidence, next time you open up some soda of any kind, pop the top, it'll fizz, it'll bubble, you leave it overnight, and what happens?
It's all gone.
It's flat. It's more or less gone. So then, why don't we feel fizzy in our bloodstream? It's under a lot of pressure, like you?
Well,
There can't be too much pressure.
It turns out there's an enzyme called carbonic anhydrase. All right, and carbonic anhydrase is responsible for the reversible hydration or dehydration of carbon dioxide. All right, it's in the red blood cells. So the carbon dioxide comes in the red blood cells, it gets hydrated, and it turns into bicarbonate, which another benefit is this is the buffering system of the bloodstream. All right, so on average of 7.4 is what the blood is. It turns out that it's actually about 7.2 in the tissue and about 7.6 in the lungs, but on average about 7.4. So we're talking about a shift of about half a pH unit.
so this is more just kind of clarification. As far as the concentration of CO2's effect on this and the Bohr's effect, does that mean like if we're more saturated with CO2, it creates more of that carbonic acid,
It
essentially
does.
because the water just converts
And
it?
essentially lowers the pH levels.
Uh-huh.
And so then the response for breathing is a response to lack of oxygen? No, buildup of CO2. Buildup of CO2. So if you want to go back and you want to do the Wim Hof thing, if you've ever looked that up, what he's encouraging is you do a series of deep breathing and inhalation, exhalation, and you do that for roughly like 30 deep breaths. When you're finished with that, what you've really done is you've exhaled most of the carbon dioxide out of your system. I mean, it's minimal at that point. And so then you can, and the funny part about it is you can just sit there for at least a minute and a half easily and not even feel the sensation of needing to breathe. Because all the CO2 has been exhaled, in a sense. If you want to try this, you can. I'm not recommending it personally. So therefore, if something goes wrong and you faint, it's not my problem, right? But yes, that's exactly, so that's one way you can actually test that out.
And so they're kind of like components that you can fluctuate yourself, as in like you can say make your body have an increase of CO2 by say holding your breath, but then you can also say fluctuate your pH levels because your body kind of has that homeostasis
That's
idea,
correct.
right?
And there are actually training regimes, if you get into physiology of exercise, there are training regimes that there are certain benefits to one or the other depending on what you're trying to do. And so there will be times where people actually induce a carbon dioxide state, so they lower the pH of their blood for an intent in training.
Wow.
Yeah, it's pretty wild.
Yeah.
So, generally speaking, so because sometimes, especially high level or elite athletes, they're looking to be able to train under conditions where they would have a high concentration of CO2 and be able to work through that and adapt to it and basically overcome the effect to some extent, so they can manage that when they're under the top conditions for racing and such.
That's called blood doping?
No, they're not blood doping. Blood doping is usually giving yourself more red blood cells, is the traditional old way of doing it. Living at high elevation also gives you the same effect. Primarily, so let's go ahead and finish out some of the story here with our hemoglobin, and we'll go look at a couple slides just for review. So the important part is, why would hemoglobin respond to this? We changed the conditions, right? pH. The question is, what in hemoglobin would respond to a change in pH? Think about your amino acids for a second. Which ones are even candidates? Histidine's one. Proline, is it ionizable? Nope. Another way to say that is, which ones have pKa's that you had to memorize? Glutamate, aspartate, arginine, lysine, cysteine, tyrosine. There we go, 7, right? Okay, so 7 of them. Of those, which one is most relevant around a pH of 7? Histidine. So we're looking for histidines. Now, we have these histidines in here. Do you think those are the ones that are relevant?
Probably.
Let's zoom back out a little bit. So, oops, that's the wrong way. All right, so if we zoom out and we make sure that the display is showing us, or settings, the display is showing us the surface. Let's just go to 20%. They're pretty covered. That's pretty tight. I mean, it's wedged in there. Oxygen get in there. So maybe a hydronium in there, but that would be really kind of weird. All right. Now, it turns out that there is such a thing on the histidine that's actually in there already. The distal histidine will hydrogen bond with the oxygen. All right. So let's go ahead and scroll around here. Oh, there it is. Okay, so this section you see here that has a little color to it. Let's go ahead and change our settings back a bit. Transparency, surface. Let's go all the way back to 50%. And let's go ahead and zoom in here for a second. It turns out that the difference between hemoglobin and myoglobin is the fact that hemoglobin has partners, quaternary structure. And the quaternary structure, what leads to the sigmoidal shape of the binding curve. Remember saw that? It's not just a hyperbolic curve, but rather it actually is a little slow, then it picks up and then it goes to saturation. All right, what's interesting is at the interface between these two, so here's the alpha subunit interfacing with this beta subunit right up here. You see that? There are a string of amino acids that are sitting right at that interface. So right here is a lysine interfacing with the carboxylic acid of the beta chain. All right, so the terminal amino acid there. All right, and then on that terminal position is a histidine, which is interfacing, so that's all on the alpha chain, is interfacing with a beta chain aspartate. There's a hydrogen bond that can form right here when histidine is protonated. So if you're just looking at it, carboxylic acid, right? pK2O, like 2-ish, right? And so, but the histidine, this one can be protonated, and when it is, there can be a hydrogen bond that forms right across here. And what do you think that's going to do if we have a quote-unquote interaction across the surface? It's going to make it tighter or looser? Tighter.
Yeah.
And what is that one binding to again?
So we're looking at an aspartate and a histidine. And that's going to make it a tighter connection between those subunits. Tighter connection between subunits is which one? T for tight, tense. So at a lower pH, going down like 7.2, this is more likely to be protonated. As it's protonated, it'll actually pull these two together, and then when that happens, it makes it more favorable to dump the oxygen off. Now, you might ask the question, how does it make it more favorable to dump the oxygen off? Watch this. This is the cool thing I wanted to show you today. So if we're looking at it a little more closely right here, if I roll it over, here's our histidine that would be locked up with the aspartate. Notice one rotation back on the same helix from the aspartate is what? Histidine in the beta subunit, and it happens to be which histidine? The proximal. Talk about a push-pull kind of system. You want to pop that oxygen off, lock it down, and you pull that back, the iron comes back, the oxygen comes off. You see that?
So that one would be the.
So this is the beta subunit. So, but it's this interface between the two right down here to where we actually lock it this way, which pulls the helix a little further back, which then actually acts on the iron, pulling it in, helping to release the oxygen atom.
So that, with the cursor on it, that's the.
That's the distal, that's the, not distal, that's the proximal
Proximal.
histamine. The proximal histamine.
So just to recap here, the oxygen itself gets pulled off because not only the proximal histidine has that pulling power, but the interaction between the aspartate and the histidine of a different part is also pulling in that same direction.
So, yes, all that's together, right?
Yeah.
All that's together in a sense, because it's constantly, if you think about it, the protein itself is just kind of happy doing its little thing, thermodynamically flexing a little bit, all right? And so what occurs though is as the pH conditions change, that's going to cause for this to be favored, which will actually favor, instead of this being a little more floppy here, it'll favor locking it in. When it pulls it in, that makes it less favorable for this because the histidine is pulling on the electron density of the iron. That's going to pull the electron density away so that it actually will disrupt the interaction on the other side. And remember, the oxygen's being held in place somewhat by the distal histidine as well. So it's got a favorable position up here, and so as you pull down, that can actually stretch that interaction. When that stretches, the electron density can flow back, and then the oxygen is freed. So I hope you can gain an appreciation how there are subtle parts of the amino acid structure in a protein that actually lead to the physical manifestations that we observe. So when Bohr saw this effect, he was not at all capable of even imagining this. They didn't even know what proteins looked like when he was doing it, right? But yet later on, it could be explained when they had structural data and further understanding. All right. The other thing, I guess I'll just briefly talk about it and then we'll switch over. So right here in the interface between these two, I think I have the other file actually, we'd have to go open that one. File, open. Should be right there. Open in a new window, okay. All right. Oh. Didn't open up here, did it?
I don't think so.
No, that one doesn't. Where did it go? Alright, well, let's do this.
Looked like it opened the second tab down the
Yeah,
bottom.
I just have to get myself straightened out. Alright, it still hasn't shown up. It's rebelling. That's gotta be it. Alright, anyway, what I was gonna point out is this interface between the beta subunits, see this clef right here? There are a series of, it's a mirrored system because they're both betas, and so it mirrors over on itself. And there are a pair of histidines, one from each subunit, a pair of lysines, one from each subunit, and there's also an amine from the terminus. All those come together giving lots of positive charge in this clef. If you have positive charge in this clef, what will it attract? Negative charge, and it turns out that 2,3-bisphosphoglycerate, think about that, 2,3-bisphospho, 2 phosphates, and a glycerate, carboxylate, that's a lot of negative charge. And so that's the molecule that actually bind in here, and it also helps to stabilize the T state. Right? And so between the bisphosphoglycerate and the pH, those are the things that help to favor that T state or release of oxygen. And that's what's actually upregulated when you go to high altitudes. How many of you have been to high altitude? Gone like up above 10,000 feet, 11,000 feet, 12,000 feet, 13, 14, 15? You're up to 15, you're like Denali or somewhere over in the Himalayas, all right? Mountaineering. So anyway, so one thing you might notice if you ever go up to high elevation is you get kind of lightheaded and you start sucking wind. Right. The best example I have is years ago, 16 years ago, I went to a conference in Breckenridge, Colorado. Been living in Montana, about the same elevation as here, so it's no different that way. Breckenridge is about 11,000 feet. Went there, hotel, everything looked nice, blah, blah, blah. Went out for a run. That lasted about 5 minutes. And I had a headache, and I just hurt. Now, by the end of the week, I went back out. I'd been walking and doing all that kind of stuff all week long. And by the end of the week, I was actually able to jog some. The question is, what happened? And the answer is 2,3-bisphosphoglycerate. That's the immediate response the body has to higher elevation adaptation, is to upregulate 2,3-bisphosphoglycerate, which is a molecule that comes almost directly out of glycolysis. So you can imagine if you're not doing you're going through glycolysis and into the citric acid cycle, but you're not doing the citric acid cycle very well because down the line you're not dumping electrons effectively to oxygen because it's lower pressure. That's going to back up. You can produce, so you can go from 1,3-bisphosphoglycerate, isomerize into 2,3-bisphosphoglycerate, and that goes in the bloodstream, and that assists in being able to bind more oxygen, deliver it, so that you can actually do oxidative phosphorylation.
The bisphosphoglycerate, what does it bind?
Oh, it binds in this. It binds in the cleft between the two beta subunits. So if I zoom back out a little bit, maybe it'll make it a little more apparent at this point. Back. So in this cleft right here is where it would be. I was trying to open the picture because I actually highlighted all those amino acids. You can see them just sticking out across that space. All right, but it does have a picture in the book. You can check it out there. So maybe we should just go there. All right, let's do that. Let's see, cursor come over. Or which way do I have to execute my screen? There it is. All right. And that stabilizes the T state. Now, if you live at a high elevation over time, you're going to end up doing what? You're going to end up actually generating more red blood cells. That's the long-term adaptation, right? And that's where blood doping comes in. So, all right, let's see, we talked about all those things. Shine light through it. There's our meat. Yes, exercise. So here's the concept of the distal versus the proximal histine, and we can see what's going on with that. Oh, by the way, if you do ever work in something like PyMol, images like this are really useful because they show you the different amino acids and their the sequence structure, and then you can figure out which one to go click on and highlight and make it show up. All right. So let's see. Yes. So this is what we're looking at here. So you can see the effects of what's different with hemoglobin. If we can actually destabilize it, what we can see is we can drop it so that normally you're going to be dropping between here for most processes. If you make it a little less lower affinity, that means for whatever you do bind, you're going to be able to drop off a little bit more. All right, that's really what's going to happen in the end. All right, so cooperative binding ligands. So there are two models people propose. They don't look that much different when you really look at it, do they? Because you start in the all relaxed state, you end in all the, or you end up in then you all end up in all the tense state, and it goes back and forth. And somebody came up with two different models, and guess what? This is the part that makes most sense. Both models fit the data. Who cares?
Those are all 4 subunits.
Yep, all 4 subunits getting filled in different permutations. So that's really what they're looking at. All right, briefly, there is a method that you can actually look at. So notice here they took the log of the concentration of oxygen, if you will, versus the, what does this look like? Some permutation of our KD thing, right? And if you plot the data this way, it's called what's called a hill plot. This is how it's useful. If there's only one binding site, notice it's a linear fit. But this is what's interesting about oxygen. It starts out doing the same exact kind of linear fit, but then at some point after one equivalent, it'll shift up and you end up at about 3 equivalents. So what it'll do is it'll actually tell you something about the number of binding sites, and it'll tell, and it's based on where the slope of the line is, it'll tell you about the affinity as well. So there is a way to analyze it mathematically. You can actually do it, you can do the titration, if you will, and observe the effect, and you can plot it, and this is how you get data for it, all right? Make it meaningful. All right, so we talked about these interactions between the subunits, all right? So with those contact points, here's the pH stuff we were talking about. You can see how the affinity changes. You should be able to make sense of this, that if you're looking at these 3 lines, that there's to be a pH, the concentration versus the binding, that it's the pH that's influencing this. All right, low pH, lower affinity. High pH, higher affinity. Tissue, blood, or lung. All right? So that's one way to look at it. And like I said, carbonic anhydrase is the enzyme responsible for taking care of this. Notice that rate increases of 5,000 times with the enzyme over just normal. And again, here's what I like. Notice it's a zinc ion that's responsible for this. So there are histidines that coordinate that zinc ion, and the zinc ion will actually bind the carbon dioxide and hydrate it. So little metal ion helping out there, just like you saw in organic chemistry, right? Put the little metal ion in there. You always put it next to the reaction arrow. didn't know what it did, you just knew, you always put it in there.
Mhm.
Yep, exactly. So
Exactly.
same thing with the enzymes. Put a little metal line in there, don't know what it does, but it still works. So, all right, so here you can see the differences between our T and our R state. Again, you can see in the blue here, they have the beta subunits in blue. You can see the space right here versus the space right there. So it's collapsed in the relaxed state, it's open in the tense state, and the BPG binds right in between here.
And that just keeps it.
That basically keeps it open in that tense state. And then what else is helping to lock it in, like we saw, were these right here, correct? The histidine aspartate. Right there. So, all right, T for taut. All right. This shows a lineup of the different interactions between the different chains. So the aspartate, histidine, carboxylate at the end, and the lysine. So these are the ones that we've looked at with this interaction right here across this space.
So the carboxylate and the lysine, that's just
It's also assisting to keep that in shape, yeah. It's basically part of your tertiary structure stabilized. All right.
So that has less to do with the actual.
Less than what this right here is the real, this one's gonna be much more sensitive than these two because the pKa values tell us that. Now, Brother Bowden told you pKa values don't always hold true, right? That's the pKa value for the free amino acid in solution, but that doesn't mean it's true if it's actually buried in a protein structure. So there can be some times that you're surprised and say, oh, okay, maybe it is now tuned to behave differently. So don't be surprised when that happens. All right, let's see, Bohr effect. We talked about the Bohr effect. We talked about these. Hopefully you saw the same thing, but I thought it was really cool because you could see the other side of the helix that's going on back here. Whereas in this picture, you really can't see it. All right, so then we have the same thing, we talked about that, there's that, and there's that, and there's that, so lots of green lines, wow, okay. And here showing us the effect, different image of it. Some Bohr effect. Anything else we really need to talk about? Oh yes, this, okay. So, transport of carbon dioxide in the blood. Most of it is transported as bicarbonate as part of the buffering system. But some of it can actually react with the terminus of our beta subunit. Interesting, huh? So we can have it actually attach to the terminus of our chains, and when that happens, it actually helps to support the T state as well. So some of it will transfer right back out this way. How much could you actually transport this way, theoretically? Up to 4 possibly, usually it's only 2 for the most part, but that's not a lot of carbon dioxide when you think about it. All right, most of it is still going to be transported in the bloodstream. But the reason why this is important is because later on this semester, when you're talking about carbohydrates specifically, you're going to see an example where carbohydrates can do something very similar and react with the terminus of the hemoglobin molecules. So Le Chatelier's principle would say if you have lots of carbohydrate, when is that happening? When you have lots of carbohydrates in your blood. Just when you've been eating something high in sugar, right? So, yeah, when you have a lot of glucose circulating around. Most of the time you eat sugar, it's sucrose, correct? Which eventually gets cleaved and you have glucose and fructose. And the fructose can be isomerized and that can turn into glucose as well, although most of it goes to the liver anyway. So next time you're down at Great Scots and you're getting something there, remember, you're loading up a lot of sugar and that can eventually attach to your hemoglobin and modify it. It's actually what they use to monitor if you really are diabetic or not. All right, so watch for that later this semester, coming soon to the Biochem class near you. All right, here's the elevation change. So BPG, here's what's really fascinating. If you had 0 BPG, it actually titrates out almost looking like myoglobin. So really the quaternary structure itself has less to do with it than the actual BPG binding and keeping it in something of the tense state. All right, that's what the data suggests. Now, the difference between when we're looking at something of 5 millimolar versus 8 millimolar, so if you're looking at 5 millimolar is normal, all right, so this would be normal kind of situation. I don't know if we're normal because we live at high elevation anyway, right?
Yeah.
So this is probably like East Coast kind of standard, all right? And then if you go to a higher elevation, so this in case, this is much higher than us. All right, so you go to higher elevation, you're gonna see that it'll shift the concentration and the graph shifts as well. We're looking at it, well, what's the difference in that? Well, if we started out here, we dropped to there, that means that now we can actually shift down. Notice how far down we end up down here. So we can actually have a shift that leads to about 37% of it being dumped off, all right? Which is different than the 30% at normal range. So we've increased the ability to dump oxygen by increasing the amount of BPG. That's what's happening. Our ability to bind it has been affected minimally, but our ability to lose it has been enhanced. All right, that can all be explained by those darn amino acids. All right. Let's see, anything else last to go? So so you can see the hole here, you can see the positive charges that I was referring to. All right, and so the BPG can actually fit in there, but when it's in the relaxed state, those get pushed together a little bit more. So if they do shift a little, so there's not so much repulsion effect. But that's what we observed happening. So you can see the BPG here, and you can see those amino acids that are surrounding it. All
So
right.
what causes BPG to fall out?
Generally, it's not falling out very much comparatively. The concentration is one thing, it's 5 millimolar. So then we'd have to look at how many molecules is that compared to how many hemoglobin molecules. But that's 5 millimolar typically in the cell. So, all right. Interesting thing to note, the alpha subunits are always there, but when when you have a fetus, as the baby's developing in the mother's womb, the baby has a problem. Mom has hemoglobin, baby has hemoglobin. How are they going to? The baby's not going to get enough oxygen if it's relying on circulating that oxygen, unless its hemoglobin can actually bind it more tightly. And so actually and before the infant's born, it actually has what's called gamma hemoglobin, so it's 2 alphas and 2 gamma subunits. These gamma subunits are expressed and they're actually missing some of the amino acid residues that are part of that binding pocket for BPG. So it binds BPG less effectively. So that means it can bind oxygen more effectively. So the baby is stealing oxygen from the mother
So it binds BPG
and hoarding
less.
it
You said it binds BPG less.
less effectively. Oxygen more. Yep. Because it doesn't have the ability to go into the tense state as well. Yeah.
Does BPG increase in pregnant women?
I don't know.
Does that help?
That's a good question. That's a good question. That'd be something definitely worth looking into. I would think not though, because she's already going to be losing enough because this situation. But that's the placental barrier to delivering the oxygen across that space, and really it makes a lot of sense because of the cellular development of the baby. You're going to be looking at a lot of cell division, all that kind of thing going on, and so there's going to be a high demand for oxygen for that energy. So, all right. And so if we're looking at it, let's see, maybe there's some information here for us. All right, so percent of total hemoglobin, so we're looking at gestational weeks. So initially it's not so high, it goes up though, and then over the 12 to 36 weeks it's pretty high. That's the gamma subunit expression, and then eventually we see that the beta subunit is increased in expression and then passes somewhere after the baby's born, a couple weeks. All right, so there's such a thing as embryonic apparently, and then it goes over the gamma for fetal, and eventually you have the adult hemoglobin. That's what's going on. Interesting enough, recently there's an actual treatment for beta thalassemia. Beta thalassemia is where there's a mutation in the beta subunit, so that it doesn't really work right. And so so as you can imagine, a child can develop and be born, and it never affects the fetal growth. But after the child starts to develop, there'll be problems with actually binding and delivering enough oxygen to the tissues. A recent advancement is with using CRISPR, they've been able to take it and treat cells, and so what they do is they treat the cells, they basically take cells and they repopulate the bone marrow is what they do, because that's where it comes out of. And so they'll actually use CRISPR to go in and downregulate the beta subunit and increase the gamma. So in other words, not trying to fix the gene per se, just change the regulation back, and then having some gamma expression allows for the individual to function more normal. So that's one of the first treatments using CRISPR that has actually been FDA approved.
Wow.
So, interesting enough.
What was it called?
Which I don't remember I don't know what they call it. It's CRISPR treatment of beta thalassemia. Just you look at that, that'll come up.
That's crazy.
So, sickle cell anemia, or sickle cell anemia. What's the origin of sickle cell? This is really interesting. What difference does a single amino acid make is the moral of the story. All right, so these are normal red blood cells. You see the nice normal cup shape. This is what happens to sickle cells. Say, what can cause such an alteration in a cell structure? And the real problem with it is normal cells can fit through the capillaries, whereas sickle cells kind of like get backed up. And this is what leads to some of the issues with sickle cell anemia, is the tissues on the extremities actually don't get enough oxygen. All right, so let's go ahead and look at what's happening here. So it turns out that the hemoglobin A can be transformed. So what happens is a glutamate at the 6th position is changed into valine. Glutamate valine. Would you say that's a similar substitution or not? So it's not conservative at all, is it? It would be considered more of a radical substitution. Now this is what can happen. And when it does happen, it turns out that you have an alteration that occurs. So if you're looking at it, this is normal. So we have hemoglobin A here, which is normal. Hemoglobin S for sickle cell. Notice there's a shape change in the beta subunit. In the beta subunit is where that mutation occurs. And specifically, what do we see? We see this. So we see this. What is called aspartate here is typically going to be there. All right, or glutamate, I mean. And it's altered in such a way that now you have now you have a valine that's sticking out in the same position. A surface valine. What do you think is going to happen? A hydrophobic amino acid at the surface. You're going to find a way to hide it, right? The reality is there's no way to fold it in because the rest of the protein is already folded in. And so what happens is it'll find a neighboring hemoglobin molecule and they will actually come together and that will sequester it between them. Problem solved, right? If it weren't for the fact that it was symmetrical. So that means it happens on each end of the hemoglobin and then on each end of that hemoglobin. And this is where we start to have this problem where they start to interact with each other, forming these chains. And so the hemoglobin starts to form these strands. And before you know it, you have a crystallization that the cell has to work around, hence the sickle shape. So when you talked earlier about, when we talked a little bit about prions and different types of proteins that can aggregate together, forming different kind of crystalline states, whether it's in the brain tissue or if it's in the hemoglobin, or it's the hemoglobin inside the red blood cells, they can form a crystalline structure, and now the cell has to adapt to that structure that's present. That's called the sickle cell. All right. So that's what we're looking at. So the valine is what we see in sickle cell as opposed to glutamic acid in normal adults. All right. Okay, so shift gears. So for the last 10 minutes, we're just going to briefly touch on the idea of a couple other structures and the relationship. I don't know how important they are to your class. I don't think they are as important as the hemoglobin. I'm pretty sure of that. Okay. All right, so another interesting example of what happens. So this is lymphocytes having binding, so they have binding proteins. So you can see different things happen with cells all the time. Red blood cells carry oxygen, platelets carry blood clotting factors, beta cells form antibodies, T cells are involved in attacking things because they have the right antibodies, and the lymphocytes, they fight individual bacteria, viruses, and fungus. Right? So we have different things that are going on to protect the body, right? So bone marrow is about 20% of blood protein, so different types of things that you can make. So all this stuff's going on. The thalamus is a primary target for controlling some of this. But ultimately, if we look at what an antibody is, it's composed of several chains of amino acids. So you'll notice here's a long continuous chain, right here in blue, and then there's a shorter chain that matches up in this area. All right, so they call one a light chain, one a heavy chain. As you can imagine, light chain, heavy chain. These are held together through disulfide bonds. All right, being out in the bloodstream, it is an oxidizing environment, so it's very common to have disulfides to help stabilize the tertiary structure. All right, and this is the part right here where all the variability happens. So in the cells, by mechanisms we will not explore at all, there are methods to randomize the amino acids that go here, and those amino acids are going to form a surface, which then will actually, if it sticks to something that's considered foreign, then that would then be tagged as something that could be attacked by the T cells, all right, or from the others. All right, so what we're looking at then is we have this ability to randomize this section and just explore and tag foreign objects. That's what antibodies are. The rest of the structure is very constant, all right? The C part represents constant variable domains right up here, just in this part, all right? And so that's what we have going on. Interesting enough, so they have at least 2 antigen binding sites. That's important to know, all right? So what'll happen is then there'll be some kind of receptor system that can then bring that thing in. The macrophages will bring it in, and they'll remember it, and they'll induce the ability to make more of these. This is what you see. What kind of secondary structure is dominant in antibodies?
Beta sheets.
Beta sheets. Beta sheets are what it mainly is. So, and then there's some carbohydrates to decorate it. But it's out here that we see is the actual part that recognizes stuff, all right? That's where we find it. So the antigen will bind to the antibody, and that's typically what will happen. Now, you can have all kinds of variations on a theme. You can look into this more if you wish in the book. I don't think the variation is important for what you need to know in the class. What's really important is, like any good biochemist or biotechnologist, what's the first thing you do when you discover something like this? You figure out, well, how can I use this to do something else? And so one of the very first things you can do is you can, well, first off, you can generate antibodies at your own will, right? By injecting something into the sheep or a rabbit or whatever it may be. You can take some of the blood out and you can get the antibodies out of the blood. Once you have those antibodies, then you can use those to do different things, all right? One of the interesting things you can end up doing is you can do a detection for something specific, whatever the antigen was, using a process called ELISA. You can also use it to tag and label structures on cells, whatever it may be. There are all sorts of things that you can do. So this idea of a high affinity antibody, we can enrich for it, and then we can use it to do different things such as library generation, biopanning, validation of different things. The way ELISA works is as follows. Typically you're going to have an antibody that you're going to so you're going to put your substrate down in the bottom here. So you're going to coat the surface with all kinds of antigens. In other words, your sample goes down there. You'll put an antibody in that would stick to the antigen of interest, and then we have an antibody that we've developed to the antibody, so it'll stick. And this antibody's been altered with some kind of an enzymatic or fluorescent capability, so we can actually detect it. And so only if our original primary antibody finds the antigen will we get a signal. So that's how you validate that it's working, all right? And so ELISA assays are really popular, because basically you can put a bunch of different samples in each of these wells, and then you put your primary antibody into each of the wells, and then you do some washing steps in between, so that if it didn't stick, it washes away. And only if it sticks will you get something that works. And so of course you have your positive and negative controls to go with it. We can see all the samples that actually had a positive, and using a spectrophotometer, you can actually measure the intensity and tell something about how much is there.
With the primary antibody, when it binds to the antigen, would it become an antigen, I guess, to the antibody?
At that point, it becomes an antigen to the next antibody, yes.
That's from
So the interesting part is, so the antibody that is the original, the primary antibody, often is made in one organism, a type of organism. So it'll be a goat, sheep, pig, whatever it may be. And then there's an entire market to generate antibodies against just antibodies from goat, pig, sheep, or whatever. And so then you just go buy your secondary antibody. You can just buy it as an anti-rabbit antibody if yours was developed in rabbit. So the first part is very specific to the antigen. The second part is specific to the organism in which it was developed.
So we have to get the 2 antigens from 2
Different animals. Yes, that's correct. So when I did some work in my PhD, developed antibodies to proteins I was studying, and I did that in rabbit. So then I just went and I bought goat antibodies, goat anti-rabbit antibodies with the tag on it, so I could then detect them. Probably the more interesting one that I did was I actually took some of the antibodies, the primary antibodies, and fused onto it gold nanoparticles of different sizes. So one had one that was 5 nanometers, and another one had one that was 10 nanometers, another one was 20 nanometers. And then I did some electron microscopy shots, so basically took bacterial cells, sliced them really thin, stuck them in the electron microscope, and then looked at, and I could see the surface of the bacterium sliced, right? You can see around the edges, and then you can see the antigen, you can see the little beads. And so I was looking for co-localization of two different proteins, and I could see the two beads were really close to each other. I couldn't see the antibodies, I could see the beads, just because the electron density of the gold is really tight, so in the electron microscope, the electrons bounce off of that really well. And so I could see those gold images of those gold beads. So there are various ways to use antibodies to get to what you want, whether it's a visualization or detection technique of any kind. There are also, they don't talk about it in here, but there are also catalytic antibodies. Next week, when we start talking about enzyme kinetics, one of the principles will be transition state theory. Remind me to talk more about that when we get there. If I'm here. If I'm not, ask Brett about it. Make him talk about it. All right. So you can also use antibodies if you're doing some kind of like protein separation using polyacrylamide gels. So you can separate your proteins, and then you can come back and you can do what's called a Western blot or an immunoblot. So you can transfer those proteins onto a different substance because it doesn't work in the gel very well. And then you can go ahead and basically block the rest of the gel with just random protein, usually something like albumin or something cheap that you can buy, milk protein, whatever. And then you'll put your primary antibody on, and it should only stick to the antigen protein. Only sticks to the antigen protein. And so at that point, you can then visualize it using different techniques as well. But again, it's a detection technique. I did that in my doctoral work as well. All right, so this just gives you some more details about what it's called. So it's Western blot. So there's that. Protein purification, if you happen to have antibodies in a column, they can just act as fish hooks to grab your particular molecule of interest. It can be a protein, it can be a small molecule, but you can just flow stuff through and you can capture it, and then you have to change the condition to allow the antibody to release it, which a change in pH can do that most of the time. Just alter the pH conditions, causes conformational shifts, and so they'll release the molecule, whatever it was. All right, and then they show some more things as terms of like the profile. Here's the flow through. Here's what you bound to the column, and it came out later. All right. Let's see, is there anything else interesting? Oh, nope, just on to myosin. So myosin is interesting in terms of muscles and how it works. Basically, we're just looking at the change here. We're focused on the idea that we have ATP binding to the head causing a conformational change. That's really the lesson we're learning from this. So muscle cells are composed of these myosin filaments that actually bind. And what will happen is all this stuff's put together. What do we want? Oh, here's a good picture. So here's the myosin head attaching to the actin filament. What we want is we want to have a movement from one place to the next. So it'll basically grab on and then slide it, right? So that's what's going to happen. And the way that it basically happens is here. All right, so we have the basic idea. So the activation site on actin is exposed as calcium 2 binds, all right? So we're going to we have ADP and PI attached to it. It's going to bind, and what's going to happen is it's going to lose the ADP and PI, and then in the process of the power stroke, and then it's going to detach, bind an ATP, and then once again it's going to go in the process of binding once again, and then it'll do the hydrolyzation, and it'll pull it through. So you just get this continuous kind of like function. Contract, release, contract, release.
Wow.
All right? And that's all instigated by binding ATP, which then gets hydrolyzed, and then that's no longer favorable for the binding event, so it detaches, and then it goes that way. All right. I guess that's all the stuff you're supposed to know. So go check those out at the end of the slide, or at the end of the slide, the presentation that he put online. When we come back on Friday, we'll focus on going over parts of the paper.